All right. Yeah.
Yes.
So thanks for
Yes.
the kind introduction and also invitation to this amazing community. I'm Saori, currently working as an instructor, but I'm very excited that I'm an incoming assistant professor at the Genome Sciences at the University of Washington in Seattle. I'm planning to start in April next year. And today I want to talk a little bit about my research on how we can integrate human genetics and single-cell genomics to define the causal mechanisms of the human diseases, especially in the autoimmune diseases field. And this is the outline of today's talk. So I want to get started with what is the current challenges in translating the disease genetic loci into the causal mechanisms of human diseases. So I think one of the interests of this group may be to define the causal mechanisms of the many human diseases and human genetics. It may seem so obvious, but human genetics is in a good position to do so because only genetics can systematically provide causal genomic loci of any disease without any hypothesis about biology, because that can tease apart the cause and consequence, complex relationship between cause and consequences. And to study the human genetic loci associated with the disease, we can use this key tool called genome-wide association study, or GWAS, which has been a quite revolutionary tool in the current human genetics field. And I think many of you are quite familiar with this, but just to get everyone on the same page, GWAS is a very simple statistical method where we can collect genotype from many cases. For example, rheumatoid arthritis patients, which is my disease of interest, and also healthy individuals, and collect many genotypes from these cases and controls. And we take just one genetic variant at a time and compare the allele frequency between cases and controls. So if the allele A is 80% in cases, but 40% in healthy individuals, we may be able to speculate that this genomic allelic variation can cause the disease risk of RA. And if you do this across the whole genome, we get this kind of representative plot called Manhattan plot because there are many buildings across the genome. So the x-axis is the chromosomal position from chromosome X to 22, and each dot is a SNP or genetic variation, and the y-axis is the significance of the disease association with RA. So as each dot or SNP becomes higher, that indicates that this SNP is strongly associated with the disease. So we published this latest GWAS result for RA, where we identified more than 100 associated loci with genome-wide significance with RA phenotype, which is great. However, the key challenge whenever we do GWAS is how to interpret a lot of disease loci. And the key point here is to consider that 90% of the risk loci that we identify from GWAS act through non-coding mechanisms. So this may also be a little obvious to you, but defining the causal mechanisms, the non-coding genome, is very challenging. So if it is a coding genome, I don't think it's an easy problem, but at least we have something like a triplet code. So if you have mutation from G to A, we can at least speculate that arginine would be changed to the lysine, which may result in some abnormal protein function that results in disease phenotype. However, in the case of the non-coding genome, we do not have yet an easily generalizable grammar regarding the consequence and impact of the genetic variant on gene regulation. For example, this allele A may regulate a very distant gene by long-range interaction in a very specific context or cell type, but this is not easily generalizable. So practically, when you want to interpret each of these 100 of loci, if you look at a single locus, so we can zoom into this single locus, we typically get this kind of situation. So again, the x-axis, the genomic position of the each SNP, each dot is a SNP or genetic variation, and the y-axis is the GWAS significance. So as we can see here, many variants are associated with the disease with the same strength. This is what is called by linkage disequilibrium structure, where we inherit these genetic variants together as a haplotype from our common ancestor. So if we merely look at the GWAS statistics, we can't distinguish a true causal variance among merely associated variants in linked disequilibrium. And also, typically, each locus contains many genes like gene A, B, C, D, and E. So even after distinguishing the true causal variant, it's not straightforward to identify the causal gene that is mediating the disease risk by having the non-coding variation at this locus. But importantly, without this causal gene information, we cannot functionally interpret this disease locus, as well as we cannot use this knowledge to develop, for example, novel therapeutic targets. So to summarize, even after we identify a lot of GWAS loci, there is a long gap from starting from the nailing down to the causal variant to the causal gene, affected cell type and tissues, affected pathway, ultimately to the disease phenotype, to truly understand the disease mechanisms. And we cannot solve this issue by using the GWAS statistics or human genetics knowledge alone. We have to add additional information to inform us to identify the causal mechanisms, and which is the functional genomics. So what do I mean by functional genomics? So we think that especially to solve the non-coding mechanisms of the GWAS locus, we think that enhancer gene maps can potentially provide the non-coding mechanisms as one layer of the functional genomics. So as you know, so we know that GWAS causal variants are highly enriched in the non-coding genome, especially in the gene regulatory regions, or what we call enhancers. So if you have a genetic variant within an enhancer, your transcription factor bindings would be slightly inhibited. That causes decreased expression of the target gene. And in this case, you can map a causal variant within an enhancer that inhibits the transcription factor binding, and also the causal gene that is being regulated by having this variant and also regulated by this functional enhancer. So once we define this enhancer gene connection, of accurate enhancer gene connection at this locus, then you can overlay with the GWAS statistics and understand or identify a single causal variant at this locus, and also the causal gene for understanding the disease mechanism. This is why we need accurate enhancer gene maps to understand the logic of the non-coding regulation at this disease risk loci. So because we understand such enhancer gene maps must be very informative, there are many previous efforts to create such enhancer gene maps from bulk tissues. For example, one of the very popular method or a conventional method that we use in the human genetics field has been EpiMap developed in the KLS lab, which correlate the epigenomic activity and gene expression at the bulk tissue level. So they used more than 800 epigenomic profiling by the ENCODE consortium to identify accurate enhancer or epigenomic and gene expression correlation to map link between the two. The other representative method in the field is the ABC model, which multiplies the epigenomic activity as putative enhancer activity, and also the physical contact between the enhancer and promoter by using the Hi-C data so that we can link the enhancer to the target gene by using the promoter as a proxy for the gene. However, conventionally, all of these methods uses bulk tissue human data, and there are challenges about that. So bulk tissue data is always described as a smoothies. So it may obscure the cell type specific nature of the gene regulation if the gene regulatory mechanism is very cell type specific, and if it's rare, you will miss by aggregating everything with other cell types. And also in a practical standpoint, it requires hundreds of samples to obtain robust association between epigenomic activation and expression variation to connect the enhancer to their target genes. So if you have an access to the large consortia like ENCODE, you can create such a genomic map for tissue-specific manner. But if you're interested in a very niche disease, when ENCODE may not necessarily have that tissue in their consortia, then you will not be able to truly use the tissue-specific, disease-specific map. But now we have, instead of the bulk tissue profiling, we now have the single cell technology. So we wondered if we can use this kind of the concept by using the single cell multimodal data to improve our resolution of the enhancer gene maps. So specifically, we use single cell multimodal ATAC-seq and RNA-seq, which can measure epigenomic activity in gene expression outcome, but at the single cell level. So obviously, like a bowl of fruit salad, you can nail down to the cell type specific nature of the gene regulation. And also, instead of doing this epigenomic and expression correlation across a few donors, you can correlate the epigenomic activity with gene expression across thousands of single cells, even when you have a few donors. So it is more democratic way to create a very tissue or cell type specific enhancer gene maps, even you have an access to tissue samples from a few donors. So in this context, we created a method called SENT, or Single Cell Enhancer Target Gene Map, and our paper is just published a few months ago in Nature Genetics in April. So we use single cell multimodal ATAC-seq and RNA-seq data, which can measure open chromatin regions, which is a putative enhancer element by ATAC-seq, and gene expression outcome by RNA-seq within individual single cells. And then we associate each ATAC-seq peak as putative enhancer element and gene pair at a time, and so to identify open chromatin regions by ATAC-seq whose activity or openness significantly correlates with gene expression outcome measured by RNA-seq as putative enhancer, functionally active enhancer gene correlation or enhancer gene lengths. So now we have the accurate enhancer gene connection, then you can map a causal variant within this functionally active enhancer at this locus, and also identify a causal gene by overlaying with the GWAS summary status, which is our ultimate goal by using this single cell genomics-based enhancer gene maps with human genetics to understand each of the genetic locus's mechanisms. But there are slightly challenging situations to develop such a method for accurate enhancer gene maps, especially in single cells. So as you can imagine, single cell data is very sparse. It doesn't, these counts doesn't normally distribute. It's very sparse, zero-skewed distribution. So the standard correlation strategy by assuming the linear regression may not work. That assumes the normal distribution of the counts. So our solution to create such method is first to use the Poisson regression, which can describe the very sparse count-based data in both gene expression and peak accessibility data. So we associated the gene expression from RNA-seq with peak accessibility with accounting for some technical covariates such as patch effect by assuming the Poisson distribution. So we essentially did the Poisson regression. However, when we did this primary analysis, we observed highly uncontrolled type 1 error or high false positive rates under now simulated dataset in the Poisson regression alone. And we did spend a lot of time figuring out what is making this statistical inflation, and figured that actually in single cell data, some of the genes are highly expressed. It's not sparsely expressed, but highly expressed, and that violates the assumption of the Poisson regression where the mean and variance should be the same. So we need to account for various distribution of the single cell multi-omics data. So we also added the bootstrapping procedure to empirically estimate the error in the coefficient in this term. So specifically, we did the primary analysis by using the full data, but followed by the bootstrapping many times to create sampled data by subsampling the cells with replacement, so that we have many empirical distribution of the data with the same regression framework. So now, after like thousands of the bootstrapping procedures, we have this beta distribution to empirically estimate how this data can be reliable and how we can estimate the empirical significance of the data or the two-sided p-values from this empirical distribution. And after we did this procedure, the QQ plot is exactly following what is expected under null. So we were happy with this calibrated statistics under the null simulation. So the final scent model, we use ATAC-seq cell by chromatin accessibility matrix and RNA-seq cell by gene expression matrix, and take one cis-peak gene pair at a time and associate gene expression with peak accessibility in a specific cell type by using Poisson regression and bootstrapping, so that we can identify significant enhancer gene connection that can be used for prioritizing causal gene and variant at the GWAS loci. So we applied our method to the real single-cell multimodal dataset, including the one from our lab, which is the RA synovial tissue from patients with rheumatoid arthritis, including a lot of inflammatory immune cells like T cells, and all the other datasets were publicly available datasets spanning from peripheral blood cells to the brain or even pituitary. And we identify more than 87,000 enhancer gene links in total, with false discovery rate under 10%. So as a recap, we tested each attack peak gene pair at a time and identified significant scent enhancer gene linkages. And as a way to understand how variable this enhancer gene links, first we plotted this coefficient beta, so to the association between peak accessibility and gene expression as a function of the distance to the transcription start site. And we found that as your enhancer get closer to the TSS, which some of them are promoters, your effect size, how strong this enhancer affects gene expression, increases, which is biologically reasonable because you expect that promoter effect is much stronger than the weaker enhancer effect on gene expression. And also, we briefly validated our scent enhancer gene links by using experimental dataset. One is CRISPR for fish, where disturbing the enhancer activity by CRISPR eye system is leading to the decreased RNA expression measured by the RNA fish, which is like how we can identify enhancer gene links by perturbing the element. And the other one is H3K27AC high chip, which is a high C combined with active enhancer promoter element by H3K27 acetylation, so that we can identify a physical link between the promoters to their enhancers. And for both of the experimental dataset, our scent enhancers were much more enriched in the positive experimentally validated enhancer gene links, meaning the odds ratio is always exceeding 1, which is very reassuring to see that our enhancer gene links are much more likely to be experimentally validated. And finally, we also wanted to know, of course, how variable our enhancer gene links are in understanding the GWAS mechanism. So we wanted to first validate if our enhancer is enriched for the known GWAS causal variants. And to identify the known GWAS causal variants, we can use the GWAS fine mapping result by statistical fine mapped variant of the large-scale biobank, such as FinGen and UK Biobank. So in FinGen, in UK Biobank, both of the large-scale biobanks, but independent, our scent enhancers in green are much more enriched in the causal GWAS variants than taking just all cis-regulatory ATAC-seq peaks alone, meaning that we were able to enrich the functionally active enhancer elements that harbors the causal variants. And also, when compared with the previous conventional enhancer gene map, such as EpiMap or ABC model, our model outperformed. And I think this result is impressive because these bulk tissue-based models uses more than 100 donors to create such maps, whereas our enhancer gene links can be created even with a single donor. But if we have thousands of single cells, which has a lot of power, has given us a lot of statistical power. Finally, I want to introduce one vignette, how it is useful in defining the disease causal variants in genes. And I'm always interested in prioritizing our own GWAS results, understanding our own GWAS results by using our own method. So we use this RA GWAS results that introduced in the first part of my presentation, and also the type 1 diabetes GWAS, so this is the chromosome 4 locus. Again, each dot is a SNP, x-axis the SNP position, and we have many, many variants associated with both of the diseases with the same strength. So we can't distinguish a true causal variance by GWAS alone, which is very challenging. However, when we used SENT, especially in T cells from rheumatoid arthritis patients, we were able to find this single enhancer, functionally active enhancer that harbors a single causal variant at this locus. And it is only observed in the inflamed T cells in RA patients, but not the other cell types. And also, more than that, we linked this causal variant to the target gene, to the RBPJ, which is the transcription factor critical for immune signaling. And the wet lab RBPJ knockdown experiment in mice resulted in abnormal regulatory T cell phenotype, making it a plausible causal gene for causing multiple autoimmune diseases. And this gene is relatively far away, 200 kb away. And if you use conventional approach to just take the closest gene, we may have misunderstood this locus by linking this variant to the other genes. So to summarize, by adding the power of the single-cell multi-omics, we started with the GWAS locus to nail down to the causal variant to the causal gene-affected cell type of the T cells, and ultimately the pathway to understand the disease mechanism of the rheumatoid arthritis. So to summarize, we created anarchic enhancer gene maps by SENT, and SENT identified enhancers with functional importance that are enriched in GWAS causal variants and outperformed previous bulk-based method, and we were able to define the causal variants in genes for the non-coding GWAS loci. And I'd like to acknowledge all of my collaborators and also the patients and participants of the donors that provided us the power to conduct such studies, and especially Shomo, currently my PI, and I'm happy to take any questions.
Thank you, Saori, for the great talk. Great job. And if there's any questions from the audience, please feel free to use the Q&A function, and then I can pass the question along. So I guess for now, a question will be, can this method sort of be applied to other regulatory elements, or it's just for enhancers?
I just call it as an enhancer, but technically it can be applied to any regulatory element. So if it's very close to the TSS, that would be the promoter. And also you can notice some of the effect of this enhancer gene links is negative, which means that it can be able to identify the repressor or silencer element. So you can flexibly identify any regulatory element that affects the gene expression in this